You know, if you've ever looked at a textbook model of a protein, you've probably wondered, what if we could actually film these atoms moving? Traditional biology is great, but it really only gives us these incredibly detailed, perfectly still photographs. The thing is, life isn't static, right? It's this continuous, super complex, microscopic dance. And if we really want to understand how life works at the absolute most fundamental level, we need a way to watch that dance unfold over time.
So, welcome to this explainer. Today, we're going to explore our ultimate high-speed molecular camera. It's called molecular dynamics simulation. This is basically the computational magic that lets researchers hit play on biology, taking those static snapshots and transforming them into dynamic, living systems.
To see how this all works, here is our quick agenda for today. We'll kick things off with a molecular video camera, then move into setting the digital stage and break down the 7-step simulation workflow. After that, we'll cover decoding the trajectory, jump into some real-world applications, and wrap up with the future of simulation. Let's get right into it.
Section 1. The molecular video camera, moving beyond static snapshots.
Okay, so what actually is this technique? Molecular dynamics simulation, or MD for short, is a computer-based method that predicts the time-dependent motion of atoms and molecules. It lets researchers observe dynamic interactions under conditions that mimic real biological environments. But here's the really fascinating part. The whole process is fundamentally driven by classical physics. You can kind of think of Newton's laws of motion as the actual script for our atomic actors. The physics calculates the forces and dictates exactly how every single atom is supposed to move on stage.
Moving on to Section 2. Setting the digital stage, gathering the cast and crew.
Look, you literally can't shoot a movie without actors and a soundstage, right? In MDA, our cast is the molecular system itself. We've got our main stars like proteins, DNA, RNA, lipids, or drug molecules, which we call ligands. But we also need our background actors to make it realistic, namely water molecules and ions. All of this gets placed inside something called the simulation box. This box perfectly mimics the physiological conditions of the human body. It is, for all intents and purposes, our digital soundstage.
But wait, how do these atomic actors actually know what to do once they're on stage? Well, they follow a force field. Now, in this context, a force field is a mathematical model that describes the interactions between atoms. We're talking about how bonds stretch, how angles bend, torsional rotations, electrostatic interactions, and even van der Waals forces. Popular force field models that scientists use, like AMBER, CHARM, OPLS, or GRAMOS, are basically the rules of physics loaded directly into the code. They tell our actors exactly how to pull, push, and bounce off of one another.
And they do this fast. Unbelievably fast. The typical time step for these physical updates is just 1 to 2 femtoseconds. To put that staggering scale into perspective, a femtosecond is 1 quadrillionth of a second. No way, right? But yes, the simulation calculates the forces and updates the positions of the atoms in these incredibly tiny increments. By stringing millions and millions of these little frames together, we generate a trajectory that spans nanoseconds or even microseconds of actual, observable biological footage.
Which brings us to Section 3, the 7-step simulation workflow. Lights, camera, action.
Let's walk through exactly how this movie gets made. We start with pre-production. Step 1 is structure preparation. We get our starting protein structure, prep any ligands, and fix any structural errors in the file. Step 2 is solvation. This is where we drop our molecule into that digital box and fill it with water molecules. And Step 3 is ion addition. We add in positive and negative ions to neutralize the system, so it mimics the real-life conditions inside a living cell. The stage is now officially set. Time to roll the cameras. Step 4 is energy minimization. This basically optimizes the structure by removing any weird, overlapping atomic contacts, like actors getting comfortable in their marks. Step 5 is equilibration, where we gradually stabilize the system's temperature and pressure. Then we get to the main event, Step 6, the production run. This is the actual MD simulation, generating the trajectory that records all those atomic movements over time. And finally, Step 7 is data analysis. We send that raw molecular footage straight to the editing room to evaluate what just happened.
Section 4. Decoding the trajectory, reviewing the footage.
So we have the footage, but how do we translate raw computational data into actual biology? Well, we use these specific parameters, kind of like a film critic's review of the performance. For example, RMSD measures the overall structural stability of our molecule during the whole run.
RMSF checks the flexibility of individual amino acid residues, so we can see exactly who is moving the most. The radius of gyration, or RG, tells us how compact our protein structure is. We also look at hydrogen bonds to check interaction stability, we check the SESA to see how much surface area was exposed to the solvent, and we calculate binding free energy to estimate just how strongly our protein and ligand interacted. This is how we mathematically prove if the simulation was a success.
Let's step out of the digital world for Section 5, Real-World Molecular Magic, from code to cures.
Why does any of this matter to us? Because this complex math has profound human outcomes. MD simulations are absolutely crucial for drug discovery. They help researchers validate early results and optimize new lead compounds. They're also used heavily in protein structure analysis to understand folding and stability. They let us uncover enzyme catalytic mechanisms, dive deep into DNA and RNA structures, and even power modern biotechnology like protein engineering. Being able to observe molecular motion in real time is a total game changer. It dramatically speeds up rational drug design and saves us from years of expensive trial and error lab work.
Now, you might be wondering what software cameras scientists are actually using to shoot these molecular blockbusters. Well, Gromox is an open-source, super fast favorite in the community. Amber is another one, hugely popular for biomolecular simulations. If you have an incredibly large system, NAMD offers amazing high-performance efficiency. Charm is a very comprehensive package, and Desmond is a high-speed commercial software that you'll see used all the time in pharmaceutical research. Actually, one really important distinction to make here is between molecular docking and molecular dynamics. People get these confused a lot. Docking is fast and static. It predicts a potential binding pose and is mostly used for virtual screening. You can think of docking as the static casting call. Molecular dynamics, on the other hand, is dynamic and heavily computationally demanding. It studies the stability of that complex over time. Dynamics is the fully realized movie that validates the findings from the initial casting call.
Which brings us to our final act, Section 6, The Future of Simulation, the sequel.
Now, for all its predictive power, I've got to be honest, this technique has some real limitations today. MD simulation is incredibly computationally intensive. It requires massive high-performance computing resources. Plus, its accuracy is strictly bound by the quality of the mathematical force fields we happen to be using. Because we demand such insane atomic detail, simulating very long biological processes can sometimes just push right past our current practical computing limits.
But the next frontier? It is absolutely thrilling. We are already looking at AI-assisted simulation analysis, enhanced sampling techniques, and integrating these simulations directly with cutting-edge cryo-electron microscopy data. Cloud-based high-performance simulations are going to democratize this massive computing power for everyone. And this paves the way for truly personalized drug discovery using a patient's own genomic info. The integration of AI and cloud computing is going to accelerate our understanding of biology and drastically speed up the development of new therapeutics.
So I want to leave you with this one final thought. As AI merges with this atomic-level simulation, unlocking our ability to perfectly model human biology on a computer with unprecedented speed, what incurable diseases will we finally be able to rewrite next? It's a pretty wild thing to think about. Thank you so much for joining me on this explainer. Keep wondering, keep learning, and I'll catch you on the next one.